The second in our two speakers this morning is Dr. Hazel Stewart, who, as I mentioned earlier, is talking about uh, SARS-CoV-2, I'll leave her to introduce the talk itself. Hazel's a senior postdoc with Andrew Firth's group in Cambridge, where she's studying gene expression mechanisms and RNA structures of positive sense RNA viruses. And I think probably, Hazel, without any further ado, I'm going to hand that over to you to take this to give us a presentation. Similarly, do please put any questions in the chat as we're ongoing, and um, and keep any questions for the end that you don't want to type. Thank you very much, Hazel.
Thanks very much, James. So hopefully everyone can hear me and see my slides. I've minimized the controls. I have an awful lot of confocal images, so I would advise other people to do the same. If you do wish to butt in during, please just shout out in case I miss the chat pop-up.
Yeah, no, Hazel, I think just take questions at the end. Don't worry about being
All right.
interrupted.
No,
Thanks.
that's fine. I was just concerned because I can't see them right now.
Yeah, don't worry.
Thank you very much. So this project was a collaboration between the Firth group, where I'm a senior postdoc, as you said, and Dr. James Edgar. So I have to give credit to James for letting us jump onto his projects. He's got a fellowship to work on Tetherin as an exosomal tether, so no virology at all. So it was definitely really enjoyable to work with an expert, and I think he enjoyed working with virologists for a change as well. So Tetherin you probably are all aware of as an antiviral restriction factor. So this protein is encoded by the BST2 gene. It's an integral membrane protein with a short N-terminal cytosolic domain, followed by a single-pass transmembrane domain, a larger extracellular coiled coil with disulfide bridges holding these parallel homodimers together. And then at the C-terminus is a short glycolipid, which acts as a membrane anchor. And as a result, this means that the protein can basically hinge itself into two separate membranes and retain or tether either extracellular vesicles or virus particles onto the cell surface. So this antiviral restriction factor is able to act upon a range of enveloped viruses. So although it was first characterized with interactions with HIV, it's been found to restrict both Ebola and various DNA viruses since. And it basically allows the host the chance to both target the budding and the envelopment process of these viruses, and therefore reducing virus release and spread, and also encourage the re-internalization and uptake and degradation of these viruses through the endosomal pathway. So as is the case with all antiviral restriction factors, viruses have obviously developed a range of multitude ways to antagonize these. So this is part of the Red Queen hypothesis with the host pathogen evolutionary arms race. And I've listed a range of Tetherin antagonists here. But generally, although they have a range of mechanisms, they tend to have the effect where they downregulate Tetherin levels on either the plasma membrane or the intracellular organelle from which the virus buds. So if we're looking at coronaviruses SARS-CoV-1 and the alpha coronavirus HCOV-2-9E were already published about 10 years ago to be restricted by Tetherin. But unlike HIV or Ebola, these don't bud from the plasma membrane. So coronaviruses bud from the ergic. This is your endoplasmic reticulum Golgi intermediate compartments. So this is a range of tubovesicular membrane clusters found between the ER and the Golgi, which allow the viruses to basically pop out of the ER. Oh, sorry, I'm getting some feedback there. They pop out of the ER and traffic across through to the cis-Golgi network out the trans end, and then traffic through vesicles to the plasma membrane. And there are two published SARS-CoV-1 antagonists before we started this work, and this was ORF3a, which was found to alter the glycosylation of Tetherin, and the spike protein, which led Tetherin to lysosomal degradation. So one thing to note is that obviously, in order to bud from the ergic and yet be restricted by Tetherin, this means that we have to see relatively high levels of Tetherin in the ergic compartments and in the secretory vesicles here, as opposed to the plasma membrane, which previous studies such as these would have focused upon. So there were three main aims to this project when we started. So first of all, does Tetherin actually restrict SARS-CoV-2? Does the virus overcome this restriction? And if so, how? Is it only the previously identified mechanisms, such as those in SARS-CoV-1, or was there something else at play? So to start with, we started doing some electron microscopy. So these are HeLa cells which constitutively express Tetherin, and we stably express ACE2 in these cells and then infect with an early lineage of SARS-CoV-2. And you can see that we did indeed see virions being tethered to the cell membrane, and they occurred in sort of polarized clusters. We could obviously see our confirmatory double membrane vesicles formed in our cells, and we could see intracellular virions stuck inside of these vesicles. When we followed this up with some immunogold staining, which is basically an EM-based Western blot, where you use antibodies labeled with gold to create these intense electron-dense particles here. So these basically are stained with an anti-Tetherin antibody. So this is reflective of a molecule or a handful of Tetherin molecules holding these viral particles to the cell surface. And this was confirmed as well with spike staining. So at first we were concerned that maybe we were just seeing extracellular vesicles as opposed to virus particles, but this indeed confirmed that Tetherin is playing a role in retaining these virus particles on the cell surface. So what if we take Tetherin away? Well, when we knock out Tetherin in our HeLa cells, both of which express ACE2, we can still see spike protein, which means that we can still see successful infection, as you would expect. But when we look at the amount of virus that actually comes out of these cells as a proportion of the total virus, so we separated intracellular and released virus at both 24 and 48 hours at a range of MOIs and looked at what percentage was released. We could see that in all cases, we saw a significant, well, not quite significant, unfortunately, according to our error bars, but a noticeable increase in how much virus was coming out in our knockout HeLa cells. So this made sense, obviously, if they're unable to hold the virus particles onto the cell surface, we should see an increase in release. So we were confident at this point that, yes, we had confirmed Tetherin does indeed play a role in restricting SARS-CoV-2. So is the virus able to overcome this restriction? So the first thing we looked at here were actually primary human cells. So these cells express very low levels of Tetherin at a steady state, as you can see here. And yet when we infect them, we see a significant increase of Tetherin expression. We would expect this given that Tetherin is an ISG. However, interestingly, we actually found that the infected cells themselves, representative here in green from spike, seem to be only neighboring the Tetherin positive cells. So this gave us an indication that Tetherin was downregulated by the presence of the virus replication itself, and yet the neighboring cells were being stimulated by the interferon release. And when we looked at a range of cell lines, this didn't quite reach statistical significance, but when we followed this up with immortalized cell lines, we saw a similar trend. So as I said, HeLa cells constitutively express Tetherin, here in red. And when we infect these in green with spike, we saw that Tetherin seemed to be excluded from the infected cells, with the neighboring cells expressing essentially similar rates of Tetherin as uninfected cells beforehand. Where we did see Tetherin coming up in our infected cells, it seemed to be more polarized towards the cell surface, and it was co-localizing with spike. So we could confirm this in other cells as well. So A549s are fairly commonly used now in SARS-CoV-2 research. They don't constitutively express Tetherin, but we can induce that expression by interferon treatment here. And when we induce Tetherin expression with interferon treatment and follow this with SARS-CoV-2 infection, we see that exclusion of Tetherin expression again in our infected cells. And when we looked at the EM of the A549 infected cells, we found an accumulation of intracellular virions, but very rare surface virions being tethered to the edges. Finally, we also looked at T84 cells. This is a colonic adenocarcinoma cell line, but it has quite a lot of microvilli, which we want to look at as it's a good epithelial example. And again, we saw that the infected cells themselves tended to have low levels of Tetherin expression, which was much higher in the neighboring adjacent cell lines. And we saw some viruses being held back at the microvilli. So we were confident that the virus was somehow able to overcome this restriction because we were seeing significantly decreased rates of Tetherin expression in infected cells themselves, even though Tetherin was actually able to restrict SARS-CoV-2. And when we knocked it out, we saw increased rates of virus release. So what mechanisms are actually at play here? So we started looking at spike because, as I said, spike has been reported in SARS-CoV-1 to downregulate Tetherin expression merely when it's on its own, so in transient and stable cell lines. So we started looking at transiently expressed tetherin with an HA tag. And as you can see here, we indeed found that even on its own, represented here in green, the HA tag, spike was able to exclude tetherin from the transfected cells. And this was confirmed again with stable cell lines using inducibly expressed spike protein. And finally, when we actually went to look at where this tetherin downregulation is occurring, we confirmed that there was a decrease in the surface levels of tetherin. So it's not just downregulating tetherin on the protein level, at a global protein level, it is decreasing the surface levels of tetherin. And this has been seen for SARS-CoV-1 spike as well. But that said, the decrease isn't really as much as we would expect, given how much we were seeing in the confocal images with our infected cells. So what other elements could be contributing to this downregulation? So when we expressed a range of our accessory proteins in transiently transfected cells and then quantified the surface levels of tetherin, we didn't actually see any significant difference from any of them. This was a bit of a surprise because, as I said, ORF3a, at least in SARS-CoV-1, has been reported to downregulate tetherin by altering the glycosylation. So we went on and looked at ORF3a anyway, just to check. And in our hands, we didn't see any downregulation of tetherin at all from either SARS-CoV-1 ORF3a or SARS-CoV-2 ORF3a. So we looked at the overall protein levels. We looked at surface levels by flow cytometry. We looked at dimerization. The
Anything for us?
A mutant is unable to form dimers. And we looked at glycosylation and couldn't see any major differences. We did see a slight difference in how punctate they seem to be with the tetherin ORF3a overlap, but it wasn't as marked as that's been published. We did, however, see that ORF3a from only SARS-CoV-1 overlapped significantly with the trans-Golgi network, and this has been published as well. It was much less so with the SARS-CoV-2 ORF3a. And we saw small, as you can see here by the white arrows, very small punctate structures seeming to occur from ORF3a, which didn't overlap with the trans-Golgi network at all. And this was quite distinct to that of the SARS-CoV-1. When we went to look at the Golgi networks in a bit more detail, we could see that the Golgi networks do indeed fragment during SARS-CoV-2 infection. So these are not transfected cells. These are infected, with both the trans and the cis Golgi being looked at. And we could see that they were basically far less reticular in both cases. And this is a similar phenotype to that which we saw in our stably expressing ORF3a cell lines. So although we didn't see any effect on tetherin, it could be that the previous literature was basically mixing up the trans-Golgi network fragmentation that we seem to be seeing with the specific effect on tetherin, because tetherin has been published to be in the Golgi at various cell lines. This is still something we're trying to unravel, but at the moment, we don't think it has a major effect on tetherin antagonism. So what about the other proteins? We looked at a range of other accessory proteins. So you can see here, ORF6, ORF8, 9b, 7a, as I said, and ORF3a. And ORF3a was the only one which really came up as an interesting overlap in our confocal experiments. So when we look at ORF3a in particular, so ORF3a is generally thought to be a viroporin, except for recent literature indicating that that's only the case in overexpression models, and that actually in infected cells, although it does still co-localize to late endosomes and lysosomes, it's more involved in the Hops complex as opposed to true viroporin activity. So that's a discussion for another day. But regardless of the system, it's agreed that it localizes to endosomes, which is exactly what we saw, these sort of endosomal-like vesicles forming. And we saw a marked rearrangement of tetherin localization to co-localize with ORF3a in these intracellular vesicles. And what we found was that ORF3a seemed to be moving tetherin away from the trans-Golgi network, where it would normally be expressed. And as I said, obviously SARS-CoV-2 has to bud from the ergic compartments, which then traffic to the TGN. So the TGN is part of the biosynthetic pathway of SARS-CoV-2 viruses. And yet 3a seems to be moving tetherin away from this area. Instead, it seems to be moving tetherin towards the endolysosomes. We could see significant increase in our tetherin overlap with our late endosomal markers. And we also saw in our electron microscopy that we saw enlarged endolysosomes in all of our ORF3a, either stable or transiently expressed cell lines. So what mechanism is this? So if ORF3a is moving Tetherin towards the endolysosomes, we at first assumed that this was due to a defective lysosomal degradation. So basically Tetherin was piling up on these lysosomes which were unable to get degraded properly. But it's possible as well that it was due to defective trafficking. So just as a reminder, generally transmembrane and secretory proteins are trafficked from the ER through the ergic into the cis-Golgi network, out the trans side, and then they travel to the cell surface. But when we talk about the steady state of a protein, we're not just talking about that initial synthesis and then maintenance. We're also talking about recycling. So recycling of secretory proteins once they're on the cell surface allows them to be moved back from the cell surface through endosomes back to the trans-Golgi and then back again to the cell surface. And this recycling of membrane proteins occurs in three main ways, which I'm not going to go into detail here. But in another paper totally unrelated to coronaviruses, they found that Tetherin mostly recycles via the retromer complex. So this retromer complex is the retrograde transport. So it moves transmembrane and membrane-associated proteins from early endosomes back to the TGN. So if we know Tetherin recycles mostly in this way, is it possible ORF3a is actually messing up the recycling process as opposed to preventing lysosomal degradation? So to look at this in more detail, we performed antibody uptake experiments. So basically in our mock cell lines, you can see that when we stain our live cells with an anti-Tetherin antibody and then fix them a few hours later and check where that antibody has gone to, we see a significant amount of the Tetherin has been moved back to the TGN. So this confirms that the Tetherin is actually being recycled via the retromer complex as we would expect, and it is moving back to the TGN through the retrograde pathway. However, when ORF3a is present in our transfected cells, we see a decrease in how much of this Tetherin antibody ended up with the TGN network. So it appears that in the presence of ORF3a, that endocytosed Tetherin is not being recovered in the way it should be. We did, however, see, although this didn't reach significance, an increase in how much of the Tetherin ended up at the late endosomes or the endolysosomal compartments. So we saw an increase in the small punctate structures with Tetherin and LAMP1 co-localization. So this confirmed that Tetherin recycling is indeed inhibited, and we think that the Tetherin is basically ending up in the wrong place where it's stuck on these endolysosomes in the presence of ORF3a. We don't think this is a specific Tetherin mechanism. When we looked at other cargo of the retromer complex, such as SIMPA, we saw a similar phenotype. So we saw that, again, the uptake of the anti-SIMPA antibody was seriously decreased in the presence of ORF3a and no longer overlapping with the TGN as we would expect. So to summarize, I hope I've convinced you that Tetherin does indeed play a role in restricting SARS-CoV-2 viruses to their cells. As we deplete Tetherin and we see an increase in virus release. However, SARS-CoV-2 appears to have evolved multiple mechanisms to downregulate Tetherin. One of these is, as previously reported, for SARS-CoV-2 through a spike-mediated mechanism, decreasing overall protein levels. The ORF7a mechanism previously published in SARS-CoV-1 wasn't reproducible in our hands, although we did see an effect on the trans and cis-Golgi network fragmentation. And we've also published the novel mechanism of ORF3a-mediated downregulation of Tetherin, where it reduces Tetherin localization in the biosynthetic organelles and redirects it to the late endolysosomes. And this work was published well, not last year now, 2023 now, with both the Firth Group and James Edgar. And I'm very happy to take questions. And thank you very much to everybody who was involved, of course.
Hazel, thank you very much indeed for a really exquisite talk. I mean, the imaging that you presented us with there is just that's a huge amount of work of the highest quality. So no, thank you very much indeed for that. I see that Dallin has already got his hand up for a question, and I'll check the chat now to see if we've got anything else. Dallin.
Thanks, James. That was really great, Hazel. Thanks so much. It was really interesting work. So can you hear me okay?
Yep, yeah. Yep.
So
just making sure I can.
I had a couple of questions. The first one was just I thought that ORF3a would hijack lysosomal trafficking to facilitate coronavirus particle egress. So it sort of fits that ORF3a would be found in those late endosomes as they're turned into lysosomes. I was just wondering whether or not you had stained in that confocal, if you've ever stained for ORF3a, Tetherin, and spike altogether.
Oh, no, we didn't.
I wonder if you can go back to your immunogold staining.
Yep.
Like right at the start, the EN.
Yep. Just checking I can. Hang on. I can't. Oh, wait, hang on. Here we go. Sorry. Apologies.
If you give us that one. Maybe did you have another image? Maybe?
Okay. Can you see my screen?
Yeah, we can.
Yeah, that's
Okay. I can't see my screen for some reason. Okay. Yes. Is this the one?
Right. Yeah. So I don't know if you can make that full screen now. That's great. Right. For me, I mean, we've done a bit in the left-hand picture. Right,
Yep.
got the inset box. For me,
Yep.
that looks like an exiting lysosome. Right. So like you've got a sort of region there. So in the boxed not in the box region, but the micrograph on the left, there's a sort of element of that looks like an exit in lysosome. I can send you some pictures of lysosomes that we've got at EM.
Yeah, definitely.
It would be interesting to do that triple staining of spike, Tetherin, and because that would fit with this figure, right, where you've got Tetherin in next to particles that are egressing from the cell.
Yep.
That's really nice. The second question was, these are obviously all bat viruses, right?
Yes.
So I just wonder if you've started looking at bat Tetherin for functionality.
That's a really good question. And because my PI is not here, I can say that no, we're not looking at bat Tetherin. And if he was here, I'd say we are. It's on my to-do list, but it's one of those things where we just have not got around to it. So we have now got some bat cell lines that we're going to apparently start looking at. And to be blunt, it's just a lack of time. But you're right. I mean, if I could start this work again, we would a whole range of different cell lines, of course.
So we had worked on ruminant Tetherin in the past, and obviously there's ruminant coronaviruses, coronaviruses, but the Tetherin gene is actually duplicated in ruminants. So there's a short and a long isoform.
Mm-hmm.
So yeah, maybe we could have a chat offline.
Yeah, definitely.
Maybe sharing some constructs, right? So you could have a look, because we're also interested in this, not in Tetherin per se, but in spike trafficking.
Yeah.
Thanks,
definitely.
Dan.
All right, thanks, James.
Wonderful. Thank you.
Hazel, there's a question from Stephen Griffin in the chat. He's asking if you pre-treated your BST2 knockout cells with interferon before infection, and whether there's a difference in titer, whether you've tried that experiment.
No, we have not tried that experiment at all. So the only BST2 knockouts we have are the HeLa cells. So we've never, at least not for this particular project, we've never treated them with interferon as well, because they naturally release enough and then we knock it out and it's gone. But that is a really good question, actually.
Stephen,
not done.
Stephen, is there any follow-up you want to add to that, or?
Sure. Yeah, I was just wondering what, you showed the difference in your released versus your intracellular virus. And nice work, by the way. Lovely. I just wondered, what proportion of the viruses are exiting through a Tetherin-containing compartment?
Yeah, so I have if I can share again, I have an incredibly confusing, just to make it worse, I have an incredibly confusing set of raw data. So I don't know if anyone else has seen this, but in the interests of this being a friendly group to talk to,
of course,
we used 3 different virus stocks, although they were all early lineage Wuhan 1. we used 3 different virus stocks in the interest of doing good science, and it was incredibly hard to get truly reproducible titers coming in and out. So this was the raw data. And when we looked at it as a percentage, we were pretty convinced. But the trends did not seem to be consistent at all. And we were wondering at some points if maybe we're even seeing a change in how much virus is able to get in. Because, I mean, as you can see, like at my initial input, I know I put the same amount on, and yet our 0 hours, we're seeing slight differences. I mean, fair enough, that's an MOI of 5, but I mean, yeah, this particular experiment was, 3 months of my life. It was an absolute nightmare.
And what do you think is going on with your intracellular virus down there at the lower MOI?
Oh, I don't know. So we basically saw that either in the knockouts, more was able to get out, or there was less piling in, but if less piling up inside. There didn't necessarily seem to be any consistency between which of those we saw. Like, I know that seems like a slight difference, but we were wondering if maybe we were losing a lot of them when we were clarifying with our cell debris. Like if things were basically stuck up inside membranes that we were losing, so it was more of a technical problem than anything else.
Yeah, I mean, have you checked that like your cells will take like fluorescent EGF or ferritin normally?
So you chipped out.
So
there.
if you supply your cells with a fluorescent EGF or ferritin or something like that, are they taking things in normally?
We did try that in the beginning, and they seemed to be then, but we didn't continue down the path. So that's a good point. I mean, that was a few months before we started it, right? But yeah, it was a question we never really answered. I think that the fact that although the trend wasn't significant, which was a depressing day, it was consistent across them all. We just thought, well, that is what it is. And there's probably something there we haven't unraveled yet.
Yeah, and sorry, one last quick question, James. How are you separating your intracellular from your secreted viruses?
I was freeze thawing the cells 3 times and then clarifying, trying to do a light clarification so that I didn't pellet too much of it away. But I did a few early experiments where we didn't clarify and simply tried to look at how much was in that sort of raw lysate, as it were, and that seemed consistent. So we continued, but
it might be worth running some gradients, but yeah, that's lovely.
Well, no, we did gradients with VLPs,
Right.
but yeah, we didn't do anything with the infected cells. That's true.
Okay, cool. I'll stop there.
Hazel, Stephen, thank you very much for a really interesting range of questions. Andrew, I think at 2 minutes past 10 we really should wrap up, but
Okay.
sorry for that. Hazel, I'm sure that you can ask Hazel questions by email afterwards, and
Yes,
but I'm sure Hazel and Craig will be very pleased to answer any questions by follow up after today. But I'd like to thank both Hazel and Craig for 2 really fascinating talks, very different and actually just perfect for this audience. Thank you both very much. And the next ICN seminar will be announced. Come through on email as you heard about this one in a few weeks' time. But no, thank you very much, and it's a great morning, a great audience. Thank you very much for really good questions as well.